So hello, everyone, and welcome. My name is Niels. I'm a PhD researcher at Ghent University and KU Leuven, which are universities that are both located in Belgium. I graduated as a veterinarian about 5 years ago, and I started my PhD shortly thereafter, first in the group of Daniel Nelson in the US, and later in the group of Yves Briers, Rob Levine, and Evelyn Meyer. And today, I will talk about my PhD research that was performed as an inter-university collaboration between these groups. During my PhD, I have focused on engineering bacteriophage-derived endolysins for the treatment of streptococcal infections that occur in the udder of dairy cows. My presentation will take about 20 to 25 minutes, and you can type your questions in the chat box and we will address them after Lona's talk. So good, let's first introduce what bovine mastitis and endolysins precisely are. Bovine mastitis is another infection that occurs in dairy cows, and it can typically be caused by a lot of different microorganisms. In my PhD research, I mainly focused on the streptococcal pathogens, which are Strep uberis, Strep disgalaxiae, and Strep agalaxiae. In total, they account for approximately 20% of all bovine mastitis cases. These infections are typically treated with antibiotics that are given intramammarily into the udder. But these streptococcal pathogens have evolved certain mechanisms to evade antibiotic therapy in the cow's udder. These are either the development of antimicrobial resistance, but they are also known to go inside the epithelial cells of the udder to hide as so-called persister cells. In addition to this, they can also form biofilms, and Strep uberis is also especially known to be able to circumvent recognition by the innate immune system of the bovine mammary epithelium, which is one of the reasons why it is the most frequently isolated streptococcal bovine mastitis pathogen. Also contributing to this is the fact that Strep uberis can replicate in the dairy stable, such as in straw, which makes it hard to eradicate Strep uberis from a dairy farm. Following this reasoning, you will see that in my research, which will soon follow, I therefore used Strep uberis as the main streptococcal bovine mastitis pathogen to be tackled. Also, governmental policies, such as, for example, the European Green Deal, aim to phase out certain critical antibiotics from animal agriculture. One example is on my slide, ceftiofur, which is still registered to treat streptococcal bovine mastitis. And because of these more stringent legislations, the dairy industry, as well as animal health companies, are now looking for alternative antimicrobials for which endolysins are proposed. These endolysins are enzymes that are produced by bacteriophages to release newly assembled virus from an infected cell at the end of the lytic replication cycle. So here you see a bacterial cell that is infected by phage. And when the virus is done replicating itself, it will start to produce the endolysins, which are here represented as orange dots. Note where the red arrow is. The endolysin is able to degrade the bacterial peptidoglycan, which results in osmotic lysis in the case of gram-positive pathogens. Indeed, there is a substantial intracellular pressure which causes the bacterial to burst or lyse. You can compare this with a balloon that is exploding after sticking it with a needle. Thus, endolysins are bacteriophage-derived enzymes that hydrolyze bacterial peptidoglycan, resulting in osmotic lysis in the case of gram-positive bacteria. If you look more in depth to the molecular structure of endolysins, then you can notice that they have a modular structure, as is shown for Plices 2 and Plices 9. These are two phage endolysins that we have previously characterized with activity against Strep uberis. As you can see, they consist of so-called EAD and CBD modules. The EADs constitute the hydrolyzing activity of the endolysin, whereas the CBDs are needed for the endolysin to bind the streptococcal cell wall. Two of my supervisors, Yves Briers and Rob Levine, they have developed a high-throughput method which is known as Versatile that allows the creation of tens of thousands engineered endolysins simultaneously by swapping the EAD and CBD modules. Discussing this versatile method in depth is a bit out of the scope of this webinar, but I refer on my slides to some papers of my colleagues Hans and Lisa, so if you want to know more about this, you can have a look at those. By means of example, I here show you some engineered endolysins that we can potentially make by versatile swapping the EAD and CBD subdomains of Plices 2 and Plices 9. So Out of the wild-type endolysin splices 2 and splices 9, we have now created 4 completely different chimeric endolysins, or in short, Cly1, Cly2, Cly3, and Cly4. This domain swapping is what we basically do in my lab, which is in principle speeding up the evolutionary process of endolysins. And I think that is actually pretty cool to do. Now that you have had a proper introduction to streptococcal bovine mastitis and bacteriophage-derived endolysins, as well as how we engineered these in my lab, let's continue with the results of my PhD research. Similar to what I showed on my previous slide, I performed domain swapping with the versatile method with 16 pre-selected EADs, 14 pre-selected CBDs, and we then created so-called libraries with these selected domains, which are here shown on the left side of this slide and are named library A to library F. You probably noticed already that these libraries contain more than just EADs and CBDs. Indeed, we also included a polyhistidine tag for purification purposes and so-called CPPs, which is the abbreviation for cell-penetrating peptide. We've used these cell-penetrating peptides to the engineered endolysins to be able to tackle the bovine streptococcal pathogens in the bovine mammary epithelium, as intracellular persistence is an important virulence factor of the streptococci to circumvent antibiotic therapy. These I told you already during the introduction. Now, if you look more closely to the libraries A to F, you'll see that the order of the EAD and CBD modules is respected in A and B, as well as in C and D, and again in E and F. But each time, the cell-penetrating peptide and the his tag are mirrored. If you know that we pre-selected 16 AADs, 14 CBDs, and 5 CPPs, you can easily multiply those numbers according to the design of each library and obtain the theoretical amount of possible combinations for each library. In total, we versatile assembled more than 88,000 engineered endolysins, which were subsequently screened for lytic activity against these bovine mastitis streptococci using a halo-based assay. This halo-based assay is shown on the right side of the slide. In this halo-based assay, we transformed E. coli with vectors that harbor these assembled engineered endolysins. If the endolysin has potent lytic activity, a clearing zone or a halo will become visible. We thereafter sequence what is inside this E. coli to see which CPP, EAD, and CBD combinations are functional in this huge pool of tens of thousands engineered endolysins. Following the identification of design rules, we then apply statistics to hit-to-lead select the most active engineered endolysins in so-called narrow-down screenings. In my PhD research, we eventually hit-to-lead selected 12 engineered endolysins that all belonged to narrow-down library A, narrow-down library C, except for one hit that belonged to library E. You can clearly see some of the design rules that we identified during our halo-based screening. For example, EAD number 1 likes to be on the second position, as it occurred in all the selected engineered endolysins except for Na2 that had EAD number 2 instead. Quite striking was that we found that the fusion of the CPP is preferred at the N-terminus, where also the size and the isoelectric point, as well as the order of the EAD and CBD subdomains, was nearly identical to the wild-type endolysin splices 2 and splices 9 that we previously characterized with activity against Strep uberis. So it's pretty cool that we speed up this evolutionary process in our lab, but let's not forget about what we can learn from nature, which already had the opportunity to select for active endolysins during thousands of years. Proceeding with these 12 selected engineered endolysins, we subsequently performed expression and purification and evaluated their bacterial killing. This was done in phosphate-buffered saline as well as in pasteurized milk against multiple strains from several streptococcal species. This was then converted to a relative in vitro scoring system, as is shown on the left side of the slide. You might see that we also challenged Staphylococcus aureus to possibly select for variants that had bispecific activity against both strepto and staphylococci. Looking at the size of the bars, you can see that the engineered endolysin NC5 was the overall best performing variant with a score of 2.1 on a scale of 3. The second and the third best performing variants were NC8 with a score of 1.5 and NC3 with a score of 1.4. In addition to this in vitro scoring system, we also performed a complementary assay that is known as a turbidity reduction assay. This is shown on the right side of the slide. This assay kinetically monitors the osmotic lysis that is caused by the endolysin. Do you remember from my introduction that I told you the bacteria were exploding like a balloon after sticking them with a needle? Well, this is what we measure with this turbidity reduction assay. The faster the curve goes down, the more potent the endolysin is. As you can see, NC5 was again the best performing engineered endolysin because its curve went down the fastest and was then followed by NC8 and NC3, again on the second and the third place. Thus, these complementary assays have the same outcome and show that NC5 was our top candidate engineered endolysin out of more than 88,000 versatile assembled variants. Next, we wanted to confirm that our top candidate engineered endolysin NC5 was indeed able to kill relevant strains of Strep ubris, Strep agalaxiae, and Strep discalaxiae. Therefore, we did similar assays as on the previous slide to test if NC5 could lyse the targeted bovine mastitis pathogens. You can see on the left panel in A that all the colored curves that represent different mastitis streptococci are going down in comparison with the negative control shown in black. This observation corroborates logarithmic reductions that were observed in a subsequent time kill assay, and these log reductions were all approximately between 1 and 4 logs. Therefore, we concluded that our selected engineered endolysin NC5 could effectively kill the bovine streptococcal pathogens that we wanted to target in my PhD research. As a side note, I should also say that we did a genetic characterization of our Strep ubris strains so that we were certain that the isolates we selected were relevant for bovine mastitis. Our isolates belong to the global clonal complexes of sequencing type 5 and 142, but regard this maybe as a detail. As I told you before, we also wanted to tackle the virulence factor of these bovine mastitis streptococci and kill them inside the bovine mammary epithelium, which we aim to do by fusing cell-penetrating peptides to the engineered endolysins. To show that our engineering strategy worked, we cultured bovine mammary epithelial cells and challenged them with our top candidate engineered endolysin NC5, after which we evaluated these cells by confocal microscopy. On the left picture of my slide, you can see these endolysin-treated cells, which have been colored blue for their nuclei, red for their actin cytoskeleton, and green for the engineered endolysin NC5. By overlaying these images, it becomes clear that NC5 is able to go inside these epithelial cells from the bovine udder. We also showed by means of Western blot that our engineered endolysin remains intact intracellularly, which made us question if our top candidate engineered endolysin could then also kill these streptococci inside these epithelial cells. On the right side of the slide, you can see that we showed for 2 bovine mammary epithelial cell lines, being the MACTs and PS cell line, that our selected endolysin NC5 is able to eradicate Strep ubris inside these cells. In my introduction, I also told you that bovine mastitis streptococci are able to form biofilms inside the udder, which allow them to circumvent antibiotic therapy. Besides being able to kill intracellularly, we also wondered if our engineering endolysin NC5 could also eradicate Strep ubris biofilm. We therefore screened our Strep ubris panel to identify strains that are able to form a biofilm and found one that did so consistently. We characterized its biofilm composition, shown in the left graph, and found it to be mainly composed of protein, as proteinase K treatment significantly eradicated this biofilm. The fact that Strep ubris biofilms are usually composed of protein is not novel, but this is just to demonstrate to you that we challenged a relevant biofilm forming strain with our engineered endolysin, as Strep ubris biofilms are always usually composed of protein. Challenging this biofilm with our top candidate engineered endolysin NC5 showed a reduction of the biofilm mass by 70%, corroborated by a reduction of approximately 1 log. Thus, NC5 is able to eradicate Strep ubris biofilm. Now we come to an important slide. which is the activity of the endolysin in raw-cast mastitic milk. We asked the Milk Control Center of Flanders to provide us with milk from Strep uberis infected dairy cows, which we subsequently challenged with our selected engineered endolysin NC5, as well as a relevant antibiotic. And we chose cloxacillin. Cloxacillin is a first-choice narrow-spectrum beta-lactam antibiotic that is frequently used to both treat and prevent Strep uberis infections in cows. It is believed to remain available for the dairy sector in the near future. We thus deliberately chose cloxacillin as it is not regarded a critical antibiotic by current governmental policies, in contrast to, for example, ceftiofur, as I told you during the introduction. When we evaluated our top candidate engineered endolysin NC5 in the mastitic milk from 4 different Strep uberis infected cows, we noticed that cloxacillin, which is represented by the orange curve, always performed better than NC5, which is the blue curve, except for cow 2 during the first 6 hours. We thus concluded that our selected endolysin as standalone therapy did not yield consistent and effective results in mastitic milk. We reasoned that this limited activity of our endolysin was caused by inhibitory compounds in the mastitic milk, such as fat and milk proteins, the presence of non-specific proteases, and Strep uberis strain differences. But in contrast to this, a very promising and key finding from our work was that we observed that a combination strategy in which cloxacillin is supplemented with our selected engineered endolysin kills all Strep uberis strains significantly faster in all mastitic milk samples. This is thus represented by the green curve. Based on this important key finding, we decided to proceed with in vivo studies in which we further investigated the potential of our selected engineered endolysin NC5, but following a combination strategy together with cloxacillin. In this mouse model, we infected lactating females in the 4th abdominal mammary gland pair with a blunted pediatric needle and inoculated a small dose of Strep uberis bacteria. In this way, we want to mimic the other infection as it occurs in the dairy cow. We then wait for 12 hours to give Strep uberis the opportunity to propagate and colonize the lactating mammary gland. We showed in preceding experiments that Strep uberis attains a growth of 1 billion colony-forming units per gram tissue at 12 hours post-inoculation, whereas we only inoculate 1,000 colony-forming units at the beginning of the experiment. When the infection is then established at 12 hours post-inoculation, we divide the mice into 4 groups. One group received a placebo treatment, another group received cloxacillin as a standalone treatment, and 2 other groups received the same dose of cloxacillin, but with the addition of either a low or a high dose of our selected engineered endolysin NC5. 4 hours after the treatment, mice were euthanized and mammary glands were harvested to look at the hallmarks of mastitis as well as other pro-inflammatory mediators. Our first observation we made when evaluating the hallmarks of mastitis was that addition of our engineered endolysin NC5 to cloxacillin dose-dependently killed Strep uberis in the mammary gland. This is here shown in A. Indeed, placebo treatment with phosphate buffered saline showed a mean of approximately 8 logs per gram tissue. But upon cloxacillin treatment, this was decreased to approximately 6.5 logs per gram tissue. Addition of our engineered endolysin NC5 to cloxacillin further decreased CFU levels by an additional half log and for the low dose and a full log for the high dose. Note that we only look 4 hours after administration of the treatment. A similar observation to the CFU levels was made for the pro-inflammatory cytokine MIP2. This is shown here in B. MIP2 is the mouse IL-8 homologue, which is a chemotactic cytokine that is produced by the infected mammary gland to recruit neutrophils from the bloodstream in order to invade the mammary gland and fight the propagating Strep uberis infection. Yes, if we look at the microscope sections of these treated mammary glands, it becomes clear that in the placebo-treated group, there is a clear and obvious influx of these neutrophils into the alveolar and ductal lumen of the infected murine mammary glands. But when the mice received treatment, there was a clear improvement of this neutrophil influx that continued to improve upon addition of our engineered endolysin NC5 to cloxacillin. Thus, we here concluded that our engineered endolysin NC5 is able to dose-dependently improve the hallmarks of mastitis in the Strep uberis infected murine mammary glands. To further support this statement, I here show you some additional pro-inflammatory mediators that all show the same trend as we observed for the hallmarks of mastitis, which were on the previous slides. Here I show you some more general pro-inflammatory mediators, such as IL-6, IL-1 beta, TNF-alpha, and IL-1 alpha. In addition, we also evaluated some other mediators that apply to macrophages, such as MCP1 and MCSF. Overall, we concluded from our in vivo studies that NC5 as an add-on to cloxacillin is able to dose-dependently reduce the hallmarks of mastitis, as well as other pro-inflammatory mediators in the Strep uberis infected murine mammary glands. We reason from our in vitro results in raw-cause mastitic milk, as well as from our in vivo results in the preclinical mouse model, that a combination strategy of cloxacillin with our versatile engineered top candidate engineered endolysin NC5 will also improve the current antibiotic treatment of cloxacillin in the Strep uberis infected cow's udder. This, however, remains to be proven and now warrants a clinical follow-up study in dairy cows. And with that, I would like to thank you for being here and listening to my work. I would also like to take this opportunity to thank all the amazing people I have worked with during the past years. Thank you for being around and always willing to help me out. I'm also very grateful for the funding that I have received during the past years, especially from the FWO. And with that, I give the floor back to you, Yves.
Thank you very much, Niels. That was very clear, but everyone who has questions, please put them in the chat box for Niels, and we will deal with them afterwards. So we will continue now first with Lonne's talk. Lonne, you can share the screen. Yeah, I confirm we can see the slide. So Lonne, please go ahead. The floor is yours. I'm very much looking forward to your talk.
Thank you, Yves and Niels, for organizing this wonderful event. I'm very honored to be here. And I'm going to talk about the work that we are doing at the University of Copenhagen in my research group that we call phage-bio. That stands for phage biology and biocontrol. And I'm going to talk to you about different ways we have developed novel phage-derived antimicrobial solutions. But overall, the goal of our research group is to identify novel molecular mechanisms of phage-bacteria interactions, but also to investigate how phages and bacteria influence each other on the evolution. And finally, what I'm focusing on today is to explore phages and their components to combat pathogenic bacteria. And so we know that the first encounter for phages when they meet their host is very specific, and that is through the interaction between receptor binding proteins. Here I showed in blue with specific host receptors, and those receptor binding proteins are very diverse among phages, and they target many different types of receptors, going from proteins and sugars and everything basically what's on the surface of bacteria. So in our research, we took the starting point to ask how we can exploit the diversity of receptor binding proteins in developing novel antimicrobials. And so we took like a different angle to the research. So not focusing on the endolysins, but rather on the receptor binding proteins. And Today I'm going to briefly introduce you to 3 different ways we did that. One is the Innolysins, where we ask, can we explore the phage RPP to allow Innolysins to kill gram-negative bacteria? We have the Pyocins, where we ask if we could use natural bacteriocins and retarget them to kill specific bacteria by using the RPPs. And then finally, the Tailosins, which is a way that we were asking, could we engineer phages into headless phages and customize their killing spectrum? So Niels already said that the gram-negative bacteria are different than the gram-positive because they have an outer membrane. And that means that the access to the peptidoglycan is masked by this outer membrane protein membrane. And therefore, we were inspired by actually what Niels showed, that some Innolysins actually are modular. They have two domains. So we thought maybe we could use the receptor binding proteins to provide specificity to Innolysins and allowing them to get in closer proximity to the host cell and bind for a longer time. And by that way, allowing them to get access to the peptidoglycan. So first we tried to prove the concept in E. coli by developing these Innolysins targeting E. coli. And for that, we used a receptor binding protein from phage T5. And we choose that because it has a C-terminal binding domain that binds irreversible to the receptor. So we thought that could be a very stable binding for the enzyme. And then we use the Innolysins also from phage T5 because it has a very limited intrinsic activity, so no activity on itself against gram-negative bacteria. So long story short, I don't want to show you all the data. It's already published, but I just want to show you these photos. And here you can see the wild type cells. And here in B, C, and D, you see examples of the Innolysin, which we in this case call EC6, causing the cells to rupture and leaking out cell material from this cytosol. And then we could also show that when we mutated the receptor, we had no activity of the Innolysin. So that means we proved that it was depending on the binding. So moving on from there, we decided to develop this system further to target. As Niels said, we work also with the bacteria that are related to the veterinary area, including food safety. And here, Campylobacter jejuni is a major problem in all countries, actually. And therefore, we decided to develop Innolysins against Campylobacter jejuni. And we used the same Innolysin, and we then used a fiber, which we predicted to be a receptor binding protein of a prophage found in Campylobacter genomes. And constructing these, we were able to show that we had killing of Campylobacter jejuni, but here you see different strains, so the effect was quite different, also depending on the Innolysins that we used. And I should say this one means that it's only part of the H fiber. So what we then decided that we could see that maybe the Innolysins with the full H fiber showed better killing, so up to, well, around 1.5 log reduction in most cases, in most strains. So we moved on with that and tested on food. So here you see chicken skin, and we just colored it for fun to see how we could distribute the liquid. But here the chicken skin were inoculated with the Campylobacter and then with the Innolysins, and then we incubated that at food storage conditions So to show that if the Innolysin could actually reduce Campylobacter on the chicken skin, and we had a nice reduction of the Campylobacter on those skins. So these enzymes are able to kill Campylobacter jejuni in foods relevant conditions. So then we move on, or this is like a current project that is just started. And in this case, we are trying to develop novel Innolysins against Salmonella. And in different countries in Africa, Salmonella typhimurium causes not only diarrhea, but also systemic infections. And in these infections, it has quite a high mortality rate. So there's an interest in targeting specific dose Salmonella typhimurium causing this kind of disease. So we are now currently developing new Innolysins, and we use a library of different Innolysins for this, and we also then use RPPs, receptor binding proteins, from a collection of phages that we have established some time ago, infecting Salmonella. So we checked the entire collection and identified phages that were able to infect these strains that originate from Africa, and saw that there were potential RPPs for this construction. And in addition, we, as Niels also was showing, we will add the cell-penetrating peptides as well to specifically target persister cells in the patients. So this work is ongoing, and I forgot, sorry, to put the name on here. It's Rayen, who just joined my group, who is responsible for that, and Athena, of course. Okay, so let's shift gears and talk about another thing that we are doing. So we are looking into these Pyocins that are naturally occurring bacteriocins that look like phage tails, and they have, or as phage, they look exactly as phage tails, as you can see here, and they have a tail, and they have a receptor binding protein, and by contracting the tail, because the tail is hollow, then it causes leakage of the cellular content and death of the cell. So these ones are found naturally targeting Pseudomonas, and it has a whole operon that is encoded by the genome, and to allow the bacteria to produce these Pyocins that normally are used for the bacteria to get rid of competing neighbors in different defined niches. So what we did, we instead of having the native tail fiber, we on a plasmid cloned. We fused the N-terminal of the fiber with the C-terminal domain of a phage receptor binding protein, and doing that, we were able. And we choose the same H fiber as we used in Innolysin, but here we use two different ones, and you can see the red amino acids are where the two fibers are different. So we were thinking those two will maybe have different host range. And we also looked into the fiber, and the variable region is what what you see here. And for what we use to fuse to the N-terminal Pyocin is this much. So what came out of this was two Pyocins that we call actually Campycins, and we showed them that they had different host range. And without going into too much detail, I can just say that for the Campycin 1, we need a chaperone for it to fold and work. And here you see the the origin of the fiber was from this strain, and it is able to kill or form clearing zone on lawns of all these strains, and the other campycin can work without the chaperone and has a complementary killing spectrum. We also showed that they are very efficient antimicrobials, more efficient than the Innolysins, I would say, because here you see that the campycin, wait, let me see that if we incubated the cells with the campycins for 3 hours, then we could have, what is this? It's like 3 to 4 log killing, and the killing was specific as we saw in the previous slide. So these campycins are very efficient antimicrobials. Then we were thinking, okay, how can we take it further? We want to now target also antibiotic resistant bacteria, and for that we have a collection of ESBL. So these are extended spectrum beta-lactamase E. coli, and using those we took a number of different samples, and we isolated 28 distinct phages, and among those were these Phapecoctaviruses that had a very broad host range. And we also, when we looked into it, we could see that it had a number of tail fibers and also some tail spikes. So what we did recently was to use these 4 different tail fibers to engineer the host range of Pyocins, and we could also show that these Pyocins had different host ranges depending on the fiber that were there, and they could kill the ESBL with up to 4 log unit reduction. So that was quite efficient as well. So the last very quick thing I will like to introduce to you is the Innolysin. So basically that is a headless phage. So it, of course, it was inspired by the Pyocin, but then we were thinking, could we go the other way around, saying instead of engineering the Pyocins, could we just take whatever phage, chop off their head, and then we then have a Innolysin or Pyocin that then has the characteristic of the phage binding with it. And we did 3 different approaches. We tried to mechanically disrupt the junction. We were inhibiting the synthesis of the capsid-related proteins, and we then also performed genetic engineering to construct a capsid-deficient phage. And it was actually possible to do so, at least number 1 and 3, but and then the phage that we used was belonging to the Akerman Vidae phages, and the reason why, which is these ones, very beautiful phages, and the reason why we choose those were they had 4 receptor binding proteins forming a complex, and each of them were able to bind and bind to specific O antigen. So we thought it would be a cool Innolysin with 4 different receptor binding proteins. So it will also these receptor binding proteins, we also showed that we could engineer them. So in that way, we would have a platform to produce Innolysin targeting 4 different O antigens at the time, whatever choice we had, depending on our engineering, to kill whatever O antigen targeting carrying bacteria we wanted. The only problem that was on our way, preventing us from having like complete success, was that the phages were transducing DNA, and that complicated the constructions a lot. So to sum up, we we developed these Innolysins to target gram-negative bacteria with endolysins by using the phage RPPs, and we developed the concept against E. coli. And we have also demonstrated developed Innolysins that target Campylobacter jejuni that can be used in food. And next, we are developing the Innolysins against Salmonella, and we also in that project are aiming at in vivo studies. For the Pyocins, we were able to show that it's possible to retarget them to kill other bacteria using phage RPPs, and particularly the Campycins are highly efficient, and we are planning to test them in live animals. Because Campylobacter colonizes the chickens, and that's the whole problem with the food safety. It would get transferred to the meat. And then we also showed recently that it's possible to engineer Pyocins to be effective towards ESBL, antibiotic-resistant E. coli. And the next thing that we would like to do is to identify the receptors for both the Campycins and the one targeting ESBL, so that we can really understand how these will work. And then for the Talosins, we demonstrated that it's possible to produce headless phages with the customized killing spectrum, but for future work, we will probably use another phage for the backbone to engineer the host recognition. And then finally, I hope you're still out there. I want to thank my group, both the current and former members, because none of this is possible without their hard work. Particular for the Campylobacter work, that is Martine Holst Sørensen, who is responsible for that, working with Athena Sampara. The Rayen is doing the Salmonella work. Anders has been working on the engineering of the Agamemnidae phages together with Cedric, and also Emra was involved in the Campylobacter work, and of course Sristi for the Pyocins targeting ESBL, and also collaborators we need, and they are listed here. And maybe you already guessed that we also use the versatile shuffling from Eve's lab. And that was it from me. Thank you for listening.
Thank you very much, Shalona. Very exciting.
Should I stop share?
Yeah, you can stop sharing. Yeah.
Okay, thank you.
Yeah, it's very exciting to see that besides the rapid developments in phage therapy, that there's also much potential in phage-inspired or phage-derived products with lysins and Innolysins and Talosins and so on. So also thank you for sharing both unpublished data. That's always cool to learn that during a webinar. I'm looking forward to read the papers as well. We got already some first questions in the meeting chats.
Yeah, I can start, Eve.
Yeah, so maybe let's start with the first question of Gopinath. He asked you, Niels, why does the chimeric Innolysin in vitro activity differs between the strains of Streptococcus uberis tested? And did you also test one concentration or more?
Well, given my, the experiments that we've done the last years, we've obviously also tested other strains than just strep and staph. And actually, this is something that we see for other pathogens as well. So not every concentration of one endolysin has the same effect on different strains. So there is always a strain-dependent effect present coming from these pathogens. So for me, the different strep uberis pathogens. And if you go in and look into literature and you go over a few endolysin papers that have tested multiple strains, then you can see that they're also there you will find this strain-dependent effect. So it's actually quite normal that some strains are less sensitive for the endolysins than others. And regarding testing multiple doses, we've been doing that for some strains, not all strains, but some. And usually you see a dose-dependent effect. So if you increase the endolysin concentration, you also get more killing.
Okay, and it's, by the way, also for every small molecule antibiotic that there's a high strain dependency. And there was another question from Charlotte. She had a very sharp view, and she asked you for the observation that with cloxacillin and versus cloxacillin and NC5 that there was that some mice exhibit a nice response and others do not. And she means in terms of bacterial load, and there are responding mice and non-responding mice. But in terms of cytokine quantification, this was less the case. Can you explain this difference?
Yes, so indeed some mice were seen as non-responders regarding the CFU load. If we then look at the cytokines, we see that there is less variation and that they all respond more or less equally. We have been looking into that in the research group as well. It's something that we do not yet fully understand, but I can come up with some hypotheses. First, again, the mice, they are genetically different. So we use outbred mice, which means that there can be a different response there towards the bacterial loads. But very recently, it has also been shown for this Streptococcus strain that it can, like I told you, hide in the mammary gland. So it is not recognized in the same way as, for example, Staph aureus or E. coli. So the bovine mammary epithelial cells, they do not recognize with their TLR2 receptors the streptococci or this specific Streptococcus strain. So it's all ongoing, and there is more and more knowledge coming towards this pathogen because it has been understudied. But I can imagine that also something happens there, that they're still able to hide in the mammary gland for some reason as a persister cells. So maybe in some mice, given that they are genetically different, they can, in some mice this might happen, while in others, they are more sensitive towards the treatments. But this is not usually the case towards Staph aureus and E. coli. So if you look at other experiments from in our group with other pathogens, we see more like a uniform response, I would say. Yes, so that's, I think, what I wanted to say. Yeah.
Thank you, Niels. Lone, Gopinath, that's also a very interesting question for you. Have you tested the frequency of bacterial resistance to Innolysin or Pyocin?
No, we haven't done that, but I can say for the Pyocins, I don't think the bacteria has a chance to develop resistance because it will get killed immediately. What can happen? It may not work if you have a population of cells with diverse receptors. So, or a population of cells where you have one mutant that doesn't allow the binding. So that would be, so I will be more concerned about the diversity than actual development of resistance. But of course, we don't know with the Innolysins because it has a different mechanism. So I think that might be different. But for the Pyocin, it's like a gunshot. You don't have a chance. So that is at least how I see it now. So yes.
Immediately that, yeah.
Yeah.
Lorna, I also have a few questions for you. So how did it feel of receiving an email in your inbox that you were elected as a hero for this webinar and that you got this invitation?
I was very touched and honored. I mean, I don't see myself as someone that could be a hero for someone. So, when I was a child, I never played football or did any sports that people could say, oh, I was really good at that. I played the piano and nobody thought that was cool. So, I felt really honored, actually. So that was wonderful. And I also been thinking about the concept of a hero, and now you defined it a bit, Yves, that a hero is someone where you read the papers and so on. Because I've been really thinking, if I have a hero, I don't think I have a current hero, because in science, and there are many reasons for that, but I had a hero when I was a Ph.D. student as a young researcher. And now you have to think back, we are in the beginning of the 90s, so things were different then. And my hero was Regine Henge Aroni, because at that time, and you probably don't know her, she's not a phage person, but maybe you know her if you studied E. coli biology, because at that time when I did my PhD, she was doing some very groundbreaking work, because she identified something that we did not know, and she explained it. And the thing was, what she looked into was stationary phase, how do cells and bacteria behave there, because most research were done on exponential growing cells. And she identified the sigma S factor and changed the way that we saw all kinds of things in the biological world. So she is my hero. But I would say later on, heroes, I think I have more heroes. Scientific heroes. I mean, science, to me, science is not enough. I more have heroes on the personal level.
Yeah.
So, yeah.
Exactly. That's what we also took up in the definition of a hero. Not only about science, but it's also inspirational on other levels. Also Niels elaborated in the beginning. Lone, there are many junior researchers here, and junior researchers, they have daily problems in the lab with the smallest practical things and field experiments. But you're now what we call a senior researcher, but once you were also a junior researcher. Do you have special advice for our junior researchers, or how was it for you being a researcher, a young researcher?
My advice would be to take your own path, because there are so many expectations, and it's impossible for anyone to fulfill them all. So you have to find a way that is your way. And that goes both for the work-life balance, as Niels were mentioned. What will fit for you in your situation as you as a person, or your living conditions with the kids or whatever. But it also goes with the science, because you have to find your own niche of own way of establish yourself as a scientist. And the thing I can recommend there is to find your own path also there. And I can say, when I was a younger scientist, I had my kids. I told my professor, who was actually the first female professor at the Technical University. So she was also kind of a role model for me. But when I was doing my postdoc, I could not work extra hours. It was not possible for me because of having 2 kids. And that was my choice at that point. So I think that is something, and you should never, even though you're a young researcher, you should not be discouraged what people are telling you, what you can do and what you cannot do. Because when I was, I don't remember if it was when I was a PhD student or postdoc, but I was told by a professor that I was not the female, so you can count it was a male, that I would not be a candidate for being a professor. So that, and what I learned from that, and that is don't believe in things like that when people tell you you can do a lot. And I think it's about finding your own path.
Thank you, Lone. That's very encouraging.
Yes.
Yeah.
And yes, yeah, I don't know. What else could I?
I think that are really very nice words and a very good advice. And I hope that all the youngsters can pick up your advice today as well. Before we close, I have just some very last aspects I would like to share with you. So this was the third webinar. It's always inspiring for me as well, and we are suddenly looking for new young researchers who would like to give the next webinar. So we help you a lot to set it up. We so that we make the barrier low. So just reach out to you, to me or your supervisor, and contact us, and we help you with that. So just thinking about who could be your hero is already an inspiring exercise. Second, I would like to invite you all for our second symposium. It's an international symposium, this time in Liège, in the south of Belgium, or the southeast of Belgium. You can register on our website. Just go to bsvm.be and click to the link of the symposium. It's also very cheap, just 25 euros. And we have some very nice speakers there. And if you want to be kept updated about all our initiatives, just become a member for free from BSVOM. Again, go to the website and you can click on membership. It's open for all Belgians or all people working with the Belgian affiliation, but in fact to everyone in the world who wants to follow up our activities. So it's for free. Just click on this website, register, so that you are in our email list. Thank you very much for attending. We were with many people, and I hope to see you next time. Bye-bye, everyone.
Thank you so much. Bye-bye.